A very good day to all the assessor. My name is Win Win and I'm going to present on my topic Helicobacter pylori infection. Okay, the name Helicobacter pylori is due to the short helical structure, if you can see here, and it's also a virulent gram-negative bacterium, as it can affect 50% of the population worldwide, as it can stay in the human host for a lifetime once one get this pathogen if you don't trade it. And then it also found mostly in the stomach pyloric region, and hence the name Helicobacter pylori. And it causes chronic gastritis, peptic ulcer disease, gastric cancer, gastric mucosa associated lymphoid tissue lymphoma. Hence, it will still continue to result in high morbidity and mortality worldwide. If you see the epidemiology map, you can see that the infection is throughout the world. In children, most newly acquired infections start before 10 years old with an overall occurring rate of 1.4% each year. There was an estimation of 4.4 billion individuals infected globally in year 2015, and the prevalence is higher in adults compared to children. Rural areas also higher due to the poor hygienic status compared to urban areas. Unfortunately, even with successful full eradication, there is also recurrence rate of 2% in developed countries and higher in developing countries of 5 to 10%. To worsen the scenario, there's also increase in the resistance rate against the mainstay treatment such as clarithromycin, metronidazole, and levofloxacin, and was observed to be high in Southeast Asia country. Fazia et al. Shows the main resistance factor is due to the mutation of the pathogen genes encoding antibiotic target proteins. Now I go to the pathogenesis. Helicobacter pylori punitive properties lies on its ability to colonize in human gastric region and establish infection in 5 major stages. Number 1, able to neutralize mucosa acidic environment via the production of urease. Number 2, move to the epithelial cells with the help of flagella. Number 3, penetrates into the epithelial cell barrier via adhesion to specific receptors such as mucin 5. Number 4, causing surrounding damage to tissue via releasing toxin and with the ability to form biofilm. And lastly, interfering metabolic processes and gastric barrier homeostasis. Virulence factor of H. pylori in soluble form can bind to the cell surface or be transported into the host cell via the host type 4 secretion system. Various effector proteins released by the pathogens includes neutrophil activating proteins, which activates neutrophils and cause cytokine production, and also WAC-A, which you can see on top, is that can damage the epithelial cell layer. And I already mentioned regarding urease and flagella. Urease, enzyme was produced by H. pylori to break down urea to carbon dioxide and ammonia via hydrolyzation. And there's other outer membrane proteins such as BAP-A, OID-A, and SAP-A are adhesive, which help the pathogen to adhere to the host cell. And CAG-A, which actually was bring into the cytoplasm through the T4SS system and causes pro-inflammatory signaling and apoptosis modulation. The CAG-A is another virulent factor whereby how it gets into the cell cytoplasm is when the pi carrying pathogen attached to the host cells. That is how the T4SS actually assists the CAG4 to go inside the cell, and its biology activity depends on the types of and numbers of the API sequence at the C terminal. There are a total of 4 types, which is API A, B, C, and D, as shown in the picture. So the CAG-A, the epithelia motifs can undergo thyroxine phosphorylation, and the C and D segment can bind to the site of the SH2 domain containing thyroxine phosphatase SHP2, which actually leads to can break the cell's adhesion and cause cellular proliferation and interleukin-8 expression. And it was also shown that with the EPIA-D motif has one of it can have 2 times gastric cancer risk due to the strong binding affinity to the SH2 compared to just only one EPIA-C motif. And 2 EPIA-C motif have higher risk of fatty ulcer disease in Asia. And the deregulation of the heat shock protein expression suppresses also the host immune response, which further allowed the escape of H. pylori from immune attack of the host and further established the infection. WAC-A is also another virulent factor which actually causes vacuolation, which is also a vacuole formation of the host cell after interfering with the epithelial cells, what it can cause is it actually causes vacuolation, cytotoxicity, and cellular apoptosis, endocytic trafficking alteration, and mitochondrial destabilization leading to cell necrosis. And the vacuoles also make the whole cell more susceptible to urease activity, and is also responsible for immunity tolerance and hence persistent infection with H. pylori via interacting with the T cells and antigen presenting cells. So this is a table to summarize the virulence factor, which I already mentioned. Urease can actually break down urea to ammonia and carbon dioxide, which neutralize the acidity in the stomach. And ammonia also can destroy the epithelium. The carbon dioxide can protect the pathogen from metabolic products harm. The flagella, it's involved in the agility and flagellin activation, which help bacteria escape from the acidic environment and protect it by displaying high motility speed. The outer membrane vesicles can internalize into the host cells, which protect the bacteria from the harm of reactive oxygen species and weaken the cellular function and induce dendritic cell activities. The Cag-Pi-A encodes T4SS, which translocate Cag-A into the cytoplasm, and then with Cag-A go through tyrosine phosphorylation, causing cellular proliferation, cell extension, and interleukin-8 expression. The WAC-A can cause epithelial cell vacuolation, leading to cell death and apoptosis, and the neutrophil activation protein causes endoplasmic reticulum stress and leads to autophagy activation and enhanced cells necrosis, and also causes innate and adaptive immune response, leading to inflammation of the gastric cell and also help protect pathogen from reactive oxygen species via biofilm formation. As mentioned earlier, there's already resistance towards standard treatment, causing reduced eradication efficacy. So innovative strategies need to be in place to improve treatment outcomes while minimizing adverse effects. A deep learning model approach by Guo et al. Actually screened through a set of more than 10,000 anti-H. pylori molecules, which successfully discovered a novel berberine derivative 8 to demonstrate effectiveness against drug-resistant strains with MIC as low as 0.25 microgram per ml. It also proved to have ideal gastric retention and show good safety profile without interfering the gut microbiota. The researcher also used activity-based protein profiling techniques to identify what is the direct target for the pathogen, which is the outer membrane protein secretion and transport system. And hence, this resulted in producing a potential new chemical entity to eradicate this infection, including those of drug-resistant strain. Targeted sonodynamic therapy is a fusion of the chitosan conjugated fucose loaded with indocyanide green. It's a newly developed nano platform which work in two mechanisms. Number one, molecular through fucus and physically through ultrasound. It uses the ultrasound to attack the plasmatic bacteria via generating singlet oxygen, and this damages the biofilms and help promote autophagy against H. pylori, including those of the multi-drug resistant strain, and it helps to preserve gut microbiota. HSRA inhibitors is also a potential new class of narrow-spectrum antibacterial agent, were shown in vivo to disrupt essential H. pylori function, including halting protein biosynthesis, impair respiratory chain, oxidative stress, and impair ATP generation. Q et al. Designed a novel multi-epitope vaccine using immunoinformatics, which selected 9 proteins of the H. pylori, which you can see in the picture, including URB, sub-A, BAP-A, and so forth, based on the virulence, antigenicity, and immunogenicity properties. And the structure show favorable affinity with toll-like receptor 2 and 4, and also the immune stimulation show robust immunogenicity. However, further in vivo experiments will be carried out to confirm the safety and efficacy of this vaccine. Covet et al. Also discovered a potential nano vaccine you use as an oral vaccine administration in the mice. Also review very good mucosal and systemic antigen specific antibody response and induce strong mucosal T helper 1 immune response via activating interferon gamma and interleukin 17R signaling. Kanzande et al. Developed a spore vector vaccine carrying H. pylori antigens urease subunit A and B found on the spore surface, administered orally in mice, also show induced T helper 17, which leads to pro-inflammatory cytokine production such as interleukin 17, 17F, and 22. However, future experiments are still required to test on addressing immune tolerance issue in H. pylori infection. Most vaccine development studies are only still at the preclinical stage. Even though it has shown protective immunity in animal models, there are still drawbacks in human hosts as due to the genetic diversity and the immune tolerance mechanism exerted by pathogen in human hosts, and hence very few vaccines make it to the human clinical trials. And the mechanism of the host response to the vaccines are still minimally known, and hence lead to a slow progression in the vaccine development. To improve efficacy of vaccine, perhaps looking into improving T cells immunity might be a crucial step, as information on antibody involvement in the immune protection against this infection is still very limited. Exosome vaccine has now risen to be a hotspot in the development on tumor vaccine, as it possesses unique advantages in antigen delivery and immunomodulation. In physiology, exosomes are released by CD4 and 8 cells to enhance immune response via strengthening B cell function. Besides, exosomes that comes from antigen-presenting cells elicit MHC1 and MHC2 on their surface, and hence are potential vaccine vectors to induce activation and proliferation of T cells. Exosomes released from mast cells also transport functional RNA and other immunomodulatory proteins, which leads to dendritic cell maturation and further proliferate T and B cells. Future research idea could consider exosome as a potential vehicle for preventive and therapeutic vaccines, as it is a nano-sized extracellular vesicular packets containing information such as proteins, miRNA, RNA, DNA, and it has good stability and exclusive biosafety and low immunogenicity, as it's a natural endogenous drug carrier present in human. So in conclusion, exosome vaccine is a potential novel vaccines candidate with the advantages of stable structure, distinct composition, and can be produced in a massive scale. These are my references. Thank you for your attention.